Hi, today I'm going to discuss with you on the COVID-19 laboratory diagnosis. And we know that in order to make a diagnosis, the doctor needs to collect a lot of information. And first of all, of course, you need based on your signs and symptoms, and also based on whether you have had close contact with someone diagnosed with COVID-19, or have you traveled to or live in any area with ongoing community spread of COVID-19. And with this data, and this actually is subjective information, and we also need to provide other evidence that you have really have direct contact or in your body this virus, evidence to virus in your body, and then just come to this laboratory diagnosis just to provide the evidence that you have experienced this virus infection. And here's the basic procedures for laboratory diagnosis. And right now, almost all the diagnosis is used to detect this virus genome. So if in your body, if we can identify virus genome, that is evidence that you have exposed to this virus. And there are a lot of press. It sounds like it's a very complicated process. Yet the entire process may be complicated, but for you as a patient, actually, it's very simple. Only thing you need to do is provide sample, and sample collection only take a few minutes. So and the rest of the work is done by the professional and the treat sample, and because they're going to detect virus genome. And usually the virus genome is very, very limited amount. So there's no machine can identify the virus genome just from your sample, but the sample needs to amplify and make more copies. And then you can use the machine to analyze to determine whether or not you have this COVID-19 virus genome, and then give you the final reports. And now let's have a look at step of this procedure. And as I mentioned earlier, that the only step you need to involve is to give you a sample, and that will take you few minutes of your time. So you need to be patient, and also don't think it's so. It's very easy procedure. There's no any danger. So at this moment, just three kinds of sample you can use for this detect the virus genomic RNA. And first one is nasal swab. So just a swab and insert into nostril, both of them. So maybe you can usually use two. One is left nostril, another one is in the right. So that gives you more chance. So you maybe use this swab twice, nasal swab. And another one is throat swab. And so just collect samples from each throat. And the third one is saliva. And so maybe this is the easiest one. There's no any. It's very simple. Just if you have saliva, just put your saliva into a cup, and then give this to doctor. So, and all three samples will be taken, and then will sent to doctor, and the second step is that the sample need to treat, because the sample we get contain a lot of debris and lot of dirty stuff. So you need to have a sample need to be treated. And so after treatment, you need to purify the virus genome. And as I mentioned, that all the assay we have right now is to detect the virus genome. That's an indirect indication that you have the viruses in your body. So after treatment, and then purify the virus genomic RNA, and next step will be put this sample into the machine, and this machine is called polymerase chain reaction machine, and the machine can amplify this virus genomic RNA. So they can amplify many times. So because original sample only contains very few copies of virus genomic RNA. So after amplification, then you can carry out this analysis. So if in your sample, if you have this virus genome, and these pieces will be amplified many times. So in the machine then can detect this different kind of virus genomes. And if you don't have, then you just have background information. If you have the genome will be amplified, you see to give a positive signal. And based on this analysis, and doctor will going to give you, if you are positive, can they give you the COVID-19 positive report. That's the report you will get. So the procedures, maybe the entire procedures, you need very highly professional people to do the entire design and analysis. But as a patient, it's very simple. You just need to spend few minutes with the trained nurse or technician, and because they need to get either nasal swab, throat swab, and you can just give the doctor the saliva, and then the doctor will going to treat sample, analyze sample, and give you the data. And the entire process is very simple, so you don't need to have any worry, and this procedure is 100% safe. There's no any danger. So if you have, it's important to diagnose early actually. If you have some symptom, and then you need to give a test, because the test is very important. Common cold, flu, or this COVID-19, and the treatment is completely different. So if you can have early diagnosis, you have a lot of advantage. So you can prepare yourself to this COVID-19 infection. And this is it for today. So if I wish to see you in my next video. Bye.